Transcription is the first step in protein synthesis. Transcription is a process of copying a sequence of DNA to produce a complementary strand of RNA. The first step of transcription begins at the start site. RNA polymerase and other proteins assemble along the DNA strand and begin to unwind the DNA strand into two separate segments. The second step of transcription starts with RNA polymerase using one strand of the DNA as a template. RNA nucleotides form complementary base pairs with the DNA template. As usual, C will pair with G. In this case, when talking about RNA, A will pair with U, meaning uracil. During DNA replication, adenosine paired with thymine. So when the RNA nucleotides are forming the complementary base pairs, whenever adenosine is on, uracil will be its base pair instead of thymine. Now the RNA strand has been fully transcribed and detaches from the DNA strand. It moves out of the nucleus into the cytoplasm and tries to find a ribosome to make a protein. The goal of translation is to translate the mRNA strand into a string of amino acids, making a protein or a polypeptide. In order to begin translating my mRNA strand into a protein, I first need to recognize the start of a gene. The start of a gene is recognized by the start codon, AUG, or also called methionine. A codon is a 3-nucleotide sequence that codes for an amino acid. If each codon is made up of 3 nucleic bases, you see here, these 3 bases makes up one codon, these make up a codon, these make up a codon, and these 3 make up a codon. This gene has 4 codons in total with 12 nucleic bases. Each codon between the start and stop codon each code for a certain amino acid. So for example, the first codon is UGG. Looking on a codon chart, you see that the codon UGG codes for the amino acid TRP. Our second codon is CCC, which looking on the codon chart, you see it codes for the amino acid PRO. We've reached the stop codon, which signals the end of the amino acid chain. So this gene has 2 amino acids. These 2 amino acids make one protein. To summarize, the central dogma describes the flow of information from DNA to RNA to protein. Protein synthesis goes in the order of DNA replication, transcription, and translation.
There are 3 types of RNA: mRNA, tRNA, and rRNA. The first type of RNA is messenger RNA, or mRNA, which is temporary copies of the information found in the DNA. Translocation is the process that converts or translates an mRNA message into a polypeptide. The second type of RNA is rRNA, or ribosomal RNA. rRNA serves as structural components of proteins, making structures known as ribosomes. The third kind of RNA is tRNA, or transfer RNA. tRNA transports amino acids to the ribosome to be assembled. In cells, tRNA forms a characteristic L shape. One end of the L has an anticodon that recognizes an mRNA codon. The other end is attached to an amino acid. A mutation is a change in an organism's DNA. Many types of mutations can occur, but typically mutations that affect a single gene happen during replication, whereas mutations that affect a group of genes or an entire chromosome happen during meiosis. Substitution is when one base is exchanged for another. The results of substitution can include a change in a codon to one that encodes a different amino acid and can cause a small change in the protein produced. Also, a change in codon to one that encodes to the same amino acid can cause no change in the protein produced. These are called silent mutations. Change in an amino acid coding to a single stop codon and cause an incomplete protein can have serious effects since the incomplete protein probably won't frame shift. In the example here, the A is substituted for a G. A point mutation is a mutation in which one nucleotide is substituted for another. That is, an incorrect nucleotide is put in the place of a correct nucleotide. Very often, such a mistake is caused and fixed by DNA polymerase. If it is not, the substitution may permanently change an organism's DNA. Another type of mutation is an insertion. An insertion is when an extra base pair is inserted into a new place in the DNA. In the example, a T is added to the sequence of DNA, which is changing the organism's gene. A deletion is when a section of DNA is lost or deleted. In this example, the A is lost or deleted. Since protein coding DNA is divided into codons 3 bases long, insertions and deletions can alter a gene so that its message is no longer correctly parsed. These changes are called frame shifts. In this example of a frame shift, a T is added to the second codon. This causes a fourth codon to be made, and this messes up the amino acid sequence. In the normal DNA sequence, GLN, PRO, and THR are produced, but when there's an insertion of T into the second codon, GLN, SER, HIS are produced, and then there's a fourth codon which doesn't code for anything. Another type of mutation is a translocation. A translocation occurs when a chromosomal rearrangement in which a segment of genetic material from one chromosome becomes heritably linked to another chromosome.